We are going live in 3, 2, 1, and go. All right. Hello and welcome everyone to the second Science Talk, which is organized by Campus Radio and the Science Club of ISA Kolkata. And today's host would be Tanisha Garg, who's a newly minted fresher from our own institute. So Tanisha, over to you. And I hope this is visible to all. Any problems, please don't shy away from writing it on in the chat box.
Thank you, Sumit. Hello, everyone, and welcome to the second talk of the Science Month organized by Campus Radio and the Science Club. Today's speaker is Valeria Eliosa.
All right. Hello, and welcome everyone to the second Science Talk.
A BS student in biomedicine from National Autonomous University of Mexico, the research institute with highest output in Mexico. She, in her high school years, got the chance to visit the Weizmann Institute for a summer camp, after which she aced her university entrance test by getting a perfect score. She joined UNAM and then went on for an internship in Star Wars Research Institute, USA, and also in IISc, India. She's working on the formation of blood cells, and she's here to talk about her BS thesis. A slight request to the audience, please post your questions at the YouTube live chat and we'll take them at the end of the talk. Now over to you, Valeria.
Thank you so much. I'm going to start sharing my screen. Okay, give me a moment. So, can you see the presentation?
Yeah, the presentation can be seen. Go ahead.
Thank you so much. Good night to you, because here is morning. But my name is Valeria Eliosa, and I'm very pleased to be here, and thanks for inviting me. I will be talking about my project, my thesis project, the project I've been working on for a while. That is called "The More the Better: Impact of Cell Density on Hematopoietic Stem and Progenitor Cells." So first of all, I'm going to tell you a little bit about my story, how I got into science. So I think everything started when I was in middle school. When I started taking the chemistry classes, I was just amazed about science. And when I was in high school in 2016, I decided to take a technical career in clinical laboratory, where I started working with fungi, with bacteria, with human blood cells, and I was just thrilled about biology. So I decided to apply for an internship in the Weizmann Institute of Science in Israel. It was only 1 month, but it was just a great experience because I started realizing how the life in a laboratory was. I started knowing how to write my notebook, how to planify my experiments. It was a really nice opportunity. And afterwards, when I came back to Mexico in 2017, I started my bachelor, which is basic biomedical research. And this is the perfect major for me because I already knew that I wanted to be a researcher and focused on biology. And my major allows us to make lab rotations every year. So in different fields, so we can know different things about different fields, different techniques. So at the end, you can choose which field you like the most. So in my first year, I was in an immunology lab. I was working with blood cells, with neutrophils specifically, and these beautiful nets they make when they attack bacteria. In my second year, I decided to take a molecular biology lab because molecular biology is everywhere. So I learned to do PCRs, Western blots, transformations. I was working with bacteria at that time, and that let me make an internship at Stowers Institute for Medical Research in Kansas City, because I basically did the same thing, the same molecular biology techniques, but in yeast. And actually, in that internship, I met one of my the biggest researchers that I admire the most, which is Dr. Alejandro Sanchez Alvarado, and he's working with planaria regeneration stem cells. So I loved the topic, and I decided to do my third rotation in stem cells lab, where I'm currently working on. And afterwards, at the beginning of 2020, I could make an internship in the Indian Institute of Science. I love it. I loved the country. I loved everything. And I could work with C. elegans embryos, which was amazing. It was a developmental biology field, and I totally loved it. So lockdown started. I barely could went out from India. I barely could come back to Mexico. But the lockdown didn't stop my scientific work, and I started a project with a couple of friends as a branch of the Public Journal Club of the developmental biology, and that's where I started reading so many papers about so many topics in the bio, and I'm just thrilled. And now I know that I want to do my PhD about some of these topics, and also the congresses and symposia I have attended is just feeding my love for science for sure. I don't know what I'm going to do next, if I'm going for a master's or a PhD, but we'll see. I'll let you know, guys. So with that said, I'm going to start talking about my project. I hope you like it and enjoy it as much as I do. And please, all the questions you have around any of these internships or the topic of my project are completely welcome. So I'll start saying that hematopoiesis is the process where all the blood cells are generated. And all this process starts with hematopoietic stem cells, which is a group of cells that have basically two characteristics. It can self-renew, which means they can maintain their population for a very, very long time, and they have the capacity to differentiate to any of the blood cell types. Hematopoietic stem cells give rise to progenitor cells, which in contrast with stem cells cannot self-renew, but they do have the capacity to differentiate to many of the blood cell types. These progenitor cells give rise to precursors, and precursors are immature cells who at the end give rise to all the mature blood cells that we know. So both stem and progenitor cells lack cell surface markers associated with differentiation, such as CD11, CD14, CD16. That's why they are called lineage negative or lin negative. And they share some markers, such as CD34, that is associated with the homing in the bone marrow where they live in adult organisms. But there are some markers that differentiate both populations. For example, CD38, that is only present in progenitor cells. So because of these two markers, and for the purpose of this study, I will be calling the stem cells as simple positive cells and the progenitor population as double positive cells. So these cells can be found in different sources. The main one, the one that we all know, is the bone marrow, followed by peripheral blood, and the most recent one, which is umbilical cord blood. Even though we have the same blood cell types, because they come from different sources, the properties they have are different, and they have different characteristics. So this study is focused on samples coming from umbilical cord blood, because this source has particular biological properties that give them an advantage over the other two sources. Just to mention some, all the cells coming from umbilical cord blood have a higher percentage of stem cells compared to the adult samples. The telomere length is longer in umbilical cord blood cells than in adult samples. Also, we can find at a higher frequency cells capable of reconstitute the entire immune system of irradiated mice in transplants in cord blood compared to bone marrow and peripheral blood. And also, they can proliferate more quickly compared to the bone marrow samples. Of course, these are just advantages. There are also disadvantages, but we're focusing only in this because also its culture and expansion in vitro has a high potential to be used for clinical treatments. So all these cells, the hematopoietic cells, can be maintained in vitro cultures using early acting cytokines in the medium, such as stem cell factor, thrombopoietin, and so on. And when we are evaluating in vitro cultures, we can measure a lot of characteristics. And I'll be focusing on two that I will be mentioning during the rest of the talk. So the first one is proliferation. Proliferation is defined as the capacity of a cell to generate daughter cells despite its maturation status, the cell type they are generating. So proliferation basically means we have a higher amount of cells at the end than what we had at the beginning. Expansion, in contrast, is the capacity of a cell to generate daughter cells that are exactly the same as the original one. What I mean, the same cell type. Just to make it clear, adding early acting cytokines to the medium of these in vitro cultures is not affecting neither proliferation nor expansion. But when we have in vitro cultures, there are so many factors that can alter our results. For example, the temperature, the medium we are using, what cells we are using, for how long we are maintaining the cells. And one of these factors is cell density. Cell density has already been proved to affect proliferation in other types of stem cells, such as neural cells or adipose tissue-derived cells. And in both, we can see that the higher the density, the higher the proliferation is. So cell density is promoting the proliferation in these two other types of stem cells, but we don't know what happens in the hematopoietic system, which leads us to the question if cell density has an impact on any of the expansion or proliferation of both population stem and progenitor cells, and if so, how is it affecting the culture? So for that, I will, when we have the samples, the umbilical cord blood samples in these funny bags, we obtain the mononuclear cells by doing a density gradient centrifugation. We take only the middle part, and then we enrich the cells of lineage negative cells using magnetic column as a negative selection. And then we select our two populations using FACS, or fluorescent activated cell sorting, and we obtain our two populations of interest, the simple positive or stem cell population, and the double positive or progenitor cells. So when we have this, we seed them in plates of 96 wells with a serum-free medium and the 4 early acting cytokines that I already mentioned in 4 different densities at 1, 10, 100, and 1,000 cells per well. And there are 3 time points that are important here. So the day 0 would be when we put the cells in the plate in different densities. At day 7, after incubation, we perform cell counting, colony-forming cell assay, and we reseed the cells to wait for another week. And at day 14, we perform the same experiments, the same protocol. So colony-forming cell assay is a process where we put some cells into a semi-solid medium of methyl cellulose. We incubate this for 2 weeks, and we evaluate the presence of these tiny dots in the medium that are colonies. Every single dot is a colony formed by many, many cells. So a colony is a group of cells. And we can classify the colonies based on their size, their color, the type of cells that are forming the colony. We can classify them as erythroid, or as myeloid, or a mix. That means that can have myeloid and erythroid cells. So this group of cells forming a colony come from the same original cell. This cell is called colony-forming cell. So one single cell can form an entire colony. And these are called CFCs, or also progenitors. The progenitors have the capacity to generate colonies. So for this, to measure proliferation, I'm going to take into account total cell number and fold increase. And to evaluate expansion, I'm going to be taking into account the colony-forming cell number. I'll explain this later. Don't worry. So first, we are evaluating the proliferation. So we have here the different densities of that we seeded the cells. We have the percentage of proliferating wells that I'm taking into account as a proliferating well, the ones that have a higher cell number at the end than at the beginning. And we have the simple positive population or stem population in red, and the progenitor population or double positive in blue. So let's see first the stem population at day 7. As we can see, the density is indeed affecting the proliferation of the cells because it's increasing the higher density is, but it's not too much. It doesn't go over 40% here. But if we compare this to the 14th day, 1 week later, this percentage increases a lot, or even reaching the 100% of the wells proliferating. So this kind of tells us that the stem cells at the first week, they are in a dormant state. So they are not reacting very quickly. They cannot proliferate a lot in the first week, but at the second week, they start proliferating a lot. They react. So they're in this dormant state at the beginning. In contrast, the progenitor cells or the double positive cells also are affected by density because the percentage also increases. But as you can see here, the proliferative potential is not changing in day 7 or at day 14. It's the same one. So this means the progenitor cells are already awake. They are already ready to proliferate in the first week, and they start proliferating at the first week. So we can conclude by this that cell density is indeed affecting proliferation. It seems that it's promoting it. We can say that stem cells are proliferating more in a slower rate, and progenitors have this constant proliferation velocity. Okay, we already know now that it is affecting proliferation, is promoting it, but how much? How much the cells are proliferating? So that we can measure it by using the fold increase that is obtained by dividing final cell number by initial cell number. Again, we have the different densities here, the fold increase here. And as we can see, in the stem population, the fold increase is not too much. It's not even passing 10%. So this means the cells, yeah, they are proliferating, but they're not proliferating a lot. For example, they're just doubling the cell number that we already had at the beginning. So for example, we have whales here that at the beginning had 10 cells, and now they have only 30 cells, which is not too much. And actually, it's not being affected by density. This is not increasing with the density. But in the case of progenitor cells or double positive, it kind of seems that it is affected by density because it increases. But at day 14, this goes crazy. It goes from 7% to over 60 times of fold increase. So this tells us that the progenitor cells are proliferating and are proliferating a lot. They have this massive cell number at day 14, at day 7 in comparison with the stem population. So we can conclude that cell density is not affecting a lot the proliferation of stem cells, but it is indeed promoting the proliferation of progenitor cells. Just to check the expansion, I told you that we are evaluating expansion by counting how many colonies this can make. So we have here again the densities and the percentage of total colonies formed. At day 7, we clearly see that the density is promoting the expansion of the stem cells. And yeah, they go over 45%. So this means stem cells are proliferating just a few, but the cells that are dividing are stem cells indeed. They are expanding. The new cells we have are stem cells. But this only happens in short term. This only happens at day 7 because at day 14, this percentage of colonies decrease a lot. So this means these cells that are proliferating at day 14 are not stem cells anymore. They are something else. They can be differentiated cells or cells that just lost the capacity to generate colonies, but they are indeed not stem cells. So they are differentiating here. In contrast with progenitor cells, this expansion potential doesn't change. It's not altered by density. And this means the progenitor cells are proliferating a lot, but they are not expanding. These new cells that are generated are not progenitor cells, are something else. But at day 14 happens the same thing. This potential decreases, this percentage decreases. And this is also telling us that, yes, at day 14, we have this massive amount of cells in these progenitor whales, but they are not mainly progenitor cells anymore. They are differentiating too. So we can conclude from here that cell density is promoting the expansion only in the stem cell population, but in short term, but in long term, it's promoting differentiation in both populations. So in summary, we can say that indeed cell density is affecting the cultures of both populations, but in different ways. So, for example, on stem cells, when we seed them, when we culture them at low densities, they don't proliferate, they don't expand. But in high densities, they don't proliferate a lot. They rather expand. They produce more stem cells in short term, but in a long term, they start differentiating. In the case of progenitor cells, at low densities, they don't proliferate nor expand, but at high densities, they do start proliferating a lot, but they are not expanding. So that means they prefer differentiation over expansion in long term also. So this leads us to so many questions because we don't know why the cells are preferring to proliferate or to expand. What is causing this? We don't know that yet. And our main goal is to know what thing on the culture is causing the cells at high densities proliferate a lot or expand a lot. And hopefully, if we can find this thing, we will try to have only one cell by adding this thing to get the same output as when we have high densities. And why is this important? Because when we are obtaining the samples from umbilical cord blood or bone marrow samples, the percentage of stem cells is very, very low. So if we want to make transplants, we need more cells. The less cells we have, the less transplants we can make. So this is a great opportunity. If we have a very small amount of cells, we can expand them in vitro and then make transplants to patients and use it for clinical treatments. So yeah, there are a lot of things we have to check. The lockdown hasn't let me continue with this, but hopefully I will start this year again. And thank you for listening. I want to thank all the members of my lab, of my current lab, my mentors in other years in the internships, and of course to the ISL Kolkata campus for inviting me. And I'm happy to take your questions.
Thank you so much, Valeria, for this really interesting talk about hematopoietic stem cells and progenitor cells. And we have questions coming up. The first question is from Alikia, and their question is, how would you know if the colony came explicitly from a single cell?
How do I know that? Oh, well, it has been measured for a very, very long time. This is completely, how do I say? Yeah, it's completely measured. We already know it's a property that they have. One single progenitor cell can generate an entire colony. That's something that is known for many, many years. You can evaluate it by, using a radioactive nucleotide. You can be sure that it's coming from the same cell. But yeah, this is something that is known for many, many years.
Great. Next question is by Abir, and they're asking, what factor causes the difference between short-term and long-term results?
What factor? I don't know. I don't know. My hypotheses are that, well, the impact of cell density is different in the other stem cell types that I mentioned before in the introduction. So for once, it is because the amount or the concentration of cytokines that is being secreted by cells. But in others is the cell-cell contact. So the cells can sense how many cells are around them, are surrounding them. So my hypothesis is that hematopoietic cells sense more the concentration of cytokines, but we don't know what cytokines or what concentration that is.
Great. So I guess, Abi, your curiosity was satisfied, but if not, then you can again type in the chat box. Next question is by Pratyay, and they are asking, does that mean cell cohesion stimulates proliferation of progenitor cells?
Does that what, sorry?
Yeah, so his question is, does cell cohesion stimulate proliferation of progenitor cells?
Well, it might be. It might be. I don't know by heart. Like I can say indeed, but I think it does.
Great. Alicia has another question, and they're asking, I understood that cell density as the initial density of the cells in the wells. Am I correct?
Yeah.
Cool. Next question is by Aisha, and they're asking, how do you factor out the ethical issues by harvesting the first set of cells from animals?
From animals? Well, it's not an invasive obtainment of the cells, because you don't hurt the animal. Or actually, I'm working with human cells. I didn't mention that, but yeah, I'm working with human samples. But in animals, it's also it's not invasive. For example, if you take the cells from the bone marrow, that is very invasive. You have to take it from the bone marrow, and it's painful. It's a little bit uncomfortable for the patients, and also for the animals, of course. But for taking umbilical cord blood samples, you don't hurt, you don't do anything to the patient. You just during the birth, you take the sample from the umbilical cord blood, and that's all.
Awesome. Then Archita has a question. Can we incorporate stem cells into bone marrow in vivo? That's the question.
Incorporate the stem cells in bone marrow in vivo? Yeah, for sure, they can. But I'm not sure what I should explain. So yeah, there are some experiments in vivo. Actually, the only way you can confirm, you can prove that what you have is a stem cell is to make transplants, because you always have to have the in vitro experiments and the in vivo experiments. So to know if it's a stem cell, you have to prove that it can self-renew, it can maintain for a very, very long time. And in vitro cultures, these experiments take months, take like 6 months. But you can also transplant the cell into mice, and they can differentiate into all the blood cell types. So you irradiate mice, and you inject or you transplant the stem cells, and if they are truly stem cells, they will regenerate the entire immune system of the mice, and the mice will survive. If they don't, then that means there weren't stem cells. I hope that answers your question.
Yeah, so I hope that answers your question, but if you have any more doubts, you can write in the chat box, or you can also email her at the email ID that she showed, and I guess she'd be happy to answer your questions. Okay. The next question is by Swagatam, and they're asking why density is affecting the expansion and proliferation rate. How do cells genetically communicate between themselves during proliferation?
Well, that's something I'm willing to know, to find out. as I said, is my hypothesis is that hematopoietic cells can sense the concentrations of the cytokines that are in the medium. But to make sure about that, I have to starve them of some of the cytokines, or if it's the cell-cell contact, I have to put membranes to avoid contact. I don't really know, but my hypothesis is that it's because of the cytokines. They are communicating by using cytokines. But I'll let you know, guys, what my results drive me.
Yep, that's a really interesting question. And Swagatam, if you're interested, then maybe you can collaborate with her to know the answer together. It would be so cool. Next question is by Aisha, and she's asking, how do you obtain the zeroth stage human subject, I guess, sample? How do you obtain that? The first sample that you obtain from the human sample.
Oh well, my laboratory is in a hospital, so there are a lot of birds in that hospital. So we have an agreement. It's an agreement. So the doctors, every time there is a birth, they collect the samples. I don't do it. I haven't been in a birth ever in my life. So they take the samples and they just give us the bags with blood, and we process the blood.
Yeah, that's really interesting. So you can be assured, Aisha, that they are not evil scientists. They always stay constant and do everything by ethics.
Yeah, for sure.
I'm just kidding, obviously. Yeah. The next question is by Alekia, and they're asking, have you observed any extreme outlier in your experiments before? And what were they? That's the question.
Well, yeah, I have. Thanks for asking. I was expecting you guys to ask me that. I don't know if you noticed that I don't have any data here in the 1000 density. That's because the methodology I was using is not letting me get this density of the stem cell population. Because as I already said, the stem cell population in samples is very, very low. So I don't get as many cells as I would want. So I had to make separated experiments where I only purify by FACS the stem population. In all the other experiments, both progenitor stem cells in every experiment come from the same source. But in these other experiments, I only had to purify the stem population, and the results are completely different. Like I told you that cell density is not promoting proliferation in stem cells. Well, in that experiment, it does. This increases massively. Like here we have like 70 times of increase at day 7, and this decreases at day 14. So the results are completely different. But I can't say a lot because it's only one experiment. I only had time to make one experiment. It might be because the methodology I'm following by FACS is affecting the cells because they stay there in this instrument for at least 20 minutes, and that might be affecting the output here. But I have to check that. I cannot say a lot. But for sure, I can say that it kind of tells me that there is a limit of cell density. It, as my title says, the more the better is not always.
Hello. we seem to have lost connection, I guess. We'll try to figure this out. Hello, Valeria. We are not able to hear you. Can you? Yeah, I guess she's rejoining. Let's wait. Yeah, let's wait for a bit. I guess she's having some issues with the connection.
Hey guys, can you hear me? Sorry, I don't know what happened.
We lost. Yeah, it's all right. So let's go back to our discussion, and we were discussing about the data, which we were talking about. That there was some outliers in your data because of the facts that you were doing.
Oh yeah, did you guys hear everything?
Yeah. I guess we did.
Okay.
All right. So if you have any more doubts, whoever asked this question, you can get back to her on her email address, or you can just write in the chat box. And we are all sorry for the delay. Yeah. So the next question. The next question is by Pratyay, and they are asking, what are the benefits of this results? As in, I guess he's asking about the medical benefits of this. So, yeah, go.
Yeah, so as I said before, the number of stem and progenitor cells that are obtained either by bone marrow or umbilical cord blood samples is very, very low. So if you want to transplant some patients, you need certain amount of cells, and it's very difficult to reach because of the low rate of cells that you get from the samples. That's why scientists are trying to expand these populations in vitro, so you can expand them in vitro. You have from this low amount of cells, you can expand them, and then you can transplant more patients. So that's the main goal, to update and to make it easier to get transplant and to be more available for patients.
Awesome. So actually, I had my own question about the data that the question was about, the outlier data. In one of your slides, you showed that in the wells with the highest density, 1,000, I guess, the stem cells, the hematopoietic stem cells, did not have any proliferation. Like it was 0. Why is that? I hope I saw it correctly.
Well, yeah, it's not that it's 0. It's just that I don't have that data, like the experiments I run for all the graphs, all the results I showed you. I never got 1,000 cells for that density for stem cells. So it's just that there's no data there. It's not there's 0. It's just there's no data. That's why I had to make other experiments separately, only purifying the stem cell population, and that's where all the results are messed up, are like completely outliers. But yeah, it's not serious. It's just that I don't have the data.
Oh, that's understandable. And yeah, it's great you did other experiments. And talking about experiments, Tita is asking, what are the further experiments you're planning to do in order to, like work on your hypothesis?
Yeah, that's a great question. So there are so many things I want to try. First, I have to be sure that the cells I'm using are viable. The survival of the survival rate of these experiments. I also want to try the differentiation, like if they are biasing towards some lineage, to lymphocytes or to granulocytes, to erythrocytes. I also want to know that. That's very important. Also, like the proportions of your population are very important. For example, that's one disadvantage of umbilical cord blood compared to bone marrow, for example, because umbilical cord blood doesn't have a lot of granulocytes, so patients are very vulnerable to infections and stuff like that while they are after the transplant. And in bone marrow samples, when you make transplants with bone marrow samples, that doesn't happen because they are biased towards this myeloid lineage. So yeah, I also want to do that, and I want to compare my data with bone marrow and peripheral blood samples, if so. But yeah, let's see what my mentor says. If he lets me make so many experiments, I'm willing to do it.
Great. And another question that is really interesting, even I'm really interested in this, is there any way you could incorporate organoids in your research or in your experiments?
Well, that's great. That's a great question. I don't know. I think it can be possible, but it is already known that hematopoietic stem cells coming from embryos are not as effective or efficient as the umbilical cord blood. So, for example, when we are embryos, stem cells are not in bone marrow. They are usually in fetal liver. But yeah, their properties are not as good as the ones coming from umbilical cord blood. So yeah, I mean, it might be possible to use organoids, but yeah, at least for my purpose, it wouldn't be that effective.
Great. So the last question of the session is a question that I guess we are all very curious to know about, is how did the pandemic actually impact your research, and just how did you work around this isolation and lockdown, and how did you complete your or work with your thesis, because a lot of students in our own institute are having this problem of not being able to work on their thesis in this certain situation. So what are your tips, or what have you done yourself to pass this hurdle?
Well, yeah, it has affected me so much because my research is based on the experiments I do, the samples I get, purification. Like all the experiments are done in the lab. I cannot run anything in my computer. So yeah, since March, I haven't been able to do any experiment, which was awful because when I came back from India, I tried to make more experiments the 2 months I could before the lockdown, and they weren't working. I don't know why, but I lost a lot. I wasted a lot of time trying to know what was happening, and then the lockdown started. So I haven't been able to continue my experiments since March of last year. That's terrible for me. But what I did was to start reading more about my area. I started reading more about the old classic papers of how to when they discovered all the stem cell population, the characteristics, all the cytokines, stuff that we just know by heart. But I wanted to know how they discovered it. And I started writing my thesis. So I think that's what has maintained me sane in this lockdown. I mean, I haven't been able to continue my research, but at least I started writing my thesis, and I have already the introduction, the methodology, and all that stuff. So I will just wait to be able to go back and add all the data. But yeah.
So that was all for this evening or morning for you, Valeria, and I hope you all like this presentation. We thank Valeria so much for being present here and telling us about her interesting research and taking all these questions and handling them perfectly. And if anyone has any more doubts, if you have any more questions, you can ask her on her email ID. And that's all for tonight, guys. And tomorrow at the same time, 8:30, we're going to have another speaker from Colombia who will talk about her research with plants. So be there. And thank you, everyone, for being present here today. And we'll be ending the stream now. Thank you so much.